Okay, I mean, we've got this test. Okay, what might test it? And again, I mean, if you can't test it, you're not in the realm of science. I mean, if there's no observations that might disconfirm it, you've got a problem. So what might test it? Well, a colleague of mine, Michael Behe, wrote a book back in 1996 called Darwin's Black Box, in which he was looking at systems like this. And what he found was he looked at the, actually inside the cell. He's a biochemist by training, and so he was looking at what he found were molecular machines inside the cells. And it's just marvelous the sorts of things that happen inside the cell. You've got self-replicating robotic manufacturing plants, information processing, storage and retrieval, signal transduction circuitry, high-efficiency, high-tech, nano-engineered motors. This got ahead of myself. Transportation and distribution systems. Automated parcel addressing, UPS labels, zip codes. I mean, things have to be delivered from one place in the cell to another. We've got all this going on inside the cell, complex monitoring and feedback control. You know, all of this in the cell and molecular biology. Now, I want you to watch a little video, which, as it were, this is state-of-the-art computer animation of what's inside the cell. And so just watch and enjoy.
This is the story of a white blood cell that finds an infection and goes through the wall of the artery in order to stop that infection. What we see now is called lipid raft. It's made up of special phospholipids that pack tightly together and hold together the membrane proteins that are specifically linked to that lipid raft. These lipid rafts move around in a phospholipid bilayer and coordinate activity. Now we are zooming down on the microvilli inside of one of those microvilli protrusions. What we see now is the polarization of actin from the actin monomers, and actin forms the cytoskeletal network. This is the Judd solvent. It cuts off a part of the actin filament. Now we see microtubulin assembling from tubular monomers. And this is called catastrophe when the microtubules move back. This is kinesin walking along the microtubule and pulling behind it a vesicle. Along this picture you can see a lot of microtubules, all with vesicles being pulled by various motor proteins. And in the center is the centrosome with its centrioles. This is the cell surface, the nucleus surface of the nuclear force, and mRNA is coming out of the nuclear force, and now it's assembling with a spliceosome that allows the mRNA to be processed before the ribosome, which is shown there, begins to read the mRNA and creates a protein. This is another ribosome assembling onto the surface of the endoplasmic reticulum or the Golgi body, and in this case, it's producing a protein that will be secreted. It's contained inside of the vesicle, inside of that membrane coat, and pulled to the outside of the cell by such a motor such as kinesin, which is shown here. These are vesicles that are being dragged from the Golgi body, which is shown right here, to the outside of the cell, and we're going to watch them being secreted into the space between the membrane and the extracellular matrix, which is above. These proteins are secreted, but they don't leave the membrane because they're bound to the membrane. And these are the proteins, the ICAM, that can reach up and bind tightly to the receptor molecules on a cell that is signaling a problem somewhere behind the cell. Once ICAM binds, it allows the white blood cell to go through a conformational change where the actin matrix dissolves and it allows it to slither into the region where it needs to eat up the bacteria or the infection.
So this is what needs to be explained. The question is whether conventional evolutionary mechanisms can do it, or whether design would be an appropriate mode of explanation to supplement conventional evolutionary explanations. So let's look at one system in particular. This is one that